Thank you for the opportunity to speak to you tonight. And it's really humbling to speak at the RI. I used to come to the RI as a child. I saw amazing science communicators like Stephen Jay Gould and Richard Dawkins speaking, and just to sort of stand virtually in their shoes is a great opportunity. And to be standing, you know, where Humphrey Davy, or in theory, where Humphrey Davy once spoke and Michael Faraday spoke, it's just incredible. And I'm so excited about it tonight. And thank you all to everyone who's come, to my family and friends and colleagues and all the people I've yet to meet. I'm ashamed I can't see you in person, but at least we all get to be wearing our slippers. So as a quick introduction, I'll just talk about what got me to be talking to you tonight and how it's going. Sorry, I'm trying to work out how to speak to at least somebody, so my own face. So at school, I was lucky enough to have some amazing science teachers, and they gave us lots of opportunities to do interesting things like blowing things up with sodium, and slightly less or more prosaic things like looking at how wood lice move around. And I moved from there to university, and I sort of kept the insect theme. And I worked on the humble fruit fly for my final year project. And whilst you may see this as a pest, actually the fruit fly has led to the 6 Nobel Prizes in the last 100 years. So it's an incredibly important scientific tool. And it was during my science undergrad that I had caught the sort of science bug truly. I was lucky enough to work in a lab with Professor Stephen Russell. And one day we were looking down a microscope, and he said, the thing you're seeing there, that's the first time anyone's ever seen that, everyone's ever made that observation. And it's that kind of moment of like, wow, that's kind of kept me going in science ever since. I then moved to Imperial College London, where I started my PhD, and I was exploring whether you can make transgenic, you can make transgenic tobacco plants that express vaccines. And the plant on the left is the one that is growing a tetanus vaccine in it. And we showed that this vaccine was able to protect against tetanus infection. And if you sort of roll the clock forward 20 years, that this work has been built on by lots of different groups. And in the last year, a group in America showed that in a large clinical trial, vaccines derived from tobacco plants are protected against infections in people. And so it's a kind of tiny little notch that I did into the kind of the path of the science. And that's the great thing about science, you're kind of building it up other areas. I then was lucky enough to set up my own group. Nobody ever really leaves Imperial. And I've been working there for the last 10 years, running a group, as Martin said, looking at respiratory infections. And so what was the road? I'm here tonight to talk about my book, Infectious, and I sort of just wanted to talk about the road to the book. So in November 2019, Dan Davis put me in touch with my now agent, Caroline, and we met up and we talked about a possible book about vaccines and antibiotics and the microbiome. And Caroline suggested this as something that people might be interested in. And I thought, well, no, I don't really have the time. You know, it sounds okay, but I'm not going to have a chance to do it. And then in March 2020, obviously the world was suddenly stood on its head, and my lab was shut down, and I had a different amount of time on my hands. And so actually that gave me the space to think about the book, but also I realized that the stuff that I'd been learning for 20 years, all these kind of facts about infectious diseases that I've been accumulating, might actually be of more interest to other people other than just me. And maybe this is a good way to share it. So 8 months of writing later, some days was good writing, other days was just grind up, and I had to get the 90,000 words onto a page. And here we are in October 2021, and the book's all written and done. And it's thanks to amazing people like Sam Carter at One World who got me through this kind of cycle to where we are. So you're here tonight to hear about the story, the book that I've written, infectious diseases and pathogens and how we fight them. So I'd like to start off with an idea, which is that nature wants you dead. And it's not just you, it wants your children and everyone you've ever met and everyone they have ever met. In fact, everyone dead. It wants you to cough and sneeze and poop yourself into an early grave. It wants your blood vessels to burst and pustules to explode all over your body. And until really recently, it was really good at this. So in the 1900s, the average age was, life expectancy was 45 years. I should already be dead, really, and potentially quite a lot of you. But thanks to many of the other people who are, thanks to these scientists and many, many others, and improvements in hygiene and the impact of modern medicine, I'm not dead, and neither of you. And we owe our extended lives to the scientists who discovered, invented, and tested new approaches to control infectious diseases. Scientists like Fleming, Ball, Jenner, Nightingale, Tu, Hillman, Sanusi, and Pasteur. And this evening, I'm going to draw on my 25 years experience as a researcher to describe how human ingenuity has led to amazing breakthroughs in controlling, preventing, and treating infectious diseases. Throughout tonight, I'm going to be talking about some of my heroes behind the breakthroughs. Some individuals, like Pasteur, have become synonymous with the identification and prevention of the microorganisms that cause infectious diseases. But others are less so. And where better to start than one of the most colourful characters I learned about while writing, who was Felix De Rel. He was a French-Canadian microbiologist, and he taught himself science and led the most remarkably colourful life. Amongst other things, he lost all his money in a failed chocolate factory. He worked all around the world in the 1920s, in a time before travel was very difficult. He was arrested by the NKVD, the predecessor of the KGB in Soviet Russia. And then a couple of months later, he was arrested by the Germans and German Wehrmacht in the Second World War. So he lived through the experiences of the 20th century. But at the same time, he also managed to discover one of the kind of important breakthroughs is that there are viruses that can actually infect bacteria. And he observed that you could kill bacteria with an extract from chicken poo. And in so doing, he discovered this bacteriophage. So he was just a kind of interesting, sort of colourful character. And he developed one of the kind of important ideas that we understand or helped us today. And whilst I focus on the individuals tonight, it's important to emphasise that science is a team sport. And this is some of the amazing people I've had the privilege to who work with me in my lab. And it's just been great working with them and kind of developing the ideas together. And they, for me, kind of reflect the many, many people that all develop and work together to make science such a great thing to do. And then just before I go on, I'm just going to apologize for a few things where I've kind of let language maybe overcome the kind of scientific things. So the first one is, nature doesn't really want you dead. Nature doesn't really want anything. It's a bit of an anthropomorphism to suggest that nature wants anything. It's more that these things happen because in spite of us. But it kind of maybe helps to think about it in the phrasing it as nature wants you dead. The second thing is that viruses are not really alive. They replicate, but they don't use all of the kind of features of like a living organism. But again, it helps me as a not virologist to think of them alive, and at least you can kill them or stop them replicating. I tend to use Latin singulars, so bacteria and data are technically plural words, but I will use bacteria and data to mean both the singular and plural. And if you don't like that, then you'll have to write your own book. And hopefully there are a few dad jokes in here. That was one that was meant to make you laugh. It's very hard to tell when there's nobody watching. So I'm just going to assume you're laughing at the bits that I think are funny. And the final thing is apologies for my cutting edge PowerPoint 1999 style slides. I've tried to cut all the yellow and blue backgrounds out, but it may sneak in. So I'm going to talk about two bits in this talk. The structure of the talk and the book really has, like all good science, there's a problem and then a solution. So in the first part, I'm going to talk about the underpinning science that helps us understand infectious diseases. And then in the second part, I'm going to talk about therapies and drugs that help us prevent and cure infectious diseases. So I'm going to briefly pause. There's a picture of Maurice Hillman to allow elder people to say, ooh, an ology. Science, as Maurice Hillman famously noted in the advert, is full of ologies, most of which are derived from Greek words. And ology comes from the Greek word logia, meaning to tell. And there are all sorts of stems that we add to ology. So we've got microbiology, which involves the study of microbes, immunology, which is how the immune system works, epidemiology, studying how diseases spread between people, and virology, the study of viruses. And it's all of these ologies that underpin our understanding of how we get infected. And by understanding how we get infected, we can then do things about it to prevent and treat ourselves. So what's an infectious disease? An infectious disease is not the disease is not the same thing as causes it. So disease refers to the symptoms. If we look here, we've got the disease, which is COVID-19, but the disease is caused by the virus, SARS-CoV-2. So they're slightly different things. The virus is the causative agent, whereas the disease is the kind of collection of the symptoms afterwards. And diseases can be both infectious and non-infectious. Infectious things is something that I could say, if I got infected, I could spread to my family and to my friends. Whereas non-infectious disease is something like a heart attack or potentially diabetes. It's something that you don't necessarily catch from another organism, can't spread onto another person. So that's the first important idea. The second important idea is we need to think about parasites. And we have to think about parasites in the context of pathogens. So the book's called Infectious, Pathogens and How We Fight Them. So pathogens are the things that we are considering, like how we're protected against them. And so different organisms, if we go right back to sort of primary school biology, different organisms have developed different tactics to acquire resources. Some capture light from the sun and grow that way, and those are the plants. And then some animals eat the plants, and we call those herbivores. And then some eat the herbivores, and those are the carnivores. So you get this kind of pathway of energy through that. But some organisms have evolved and have evolved down a different path, which means instead of capturing energy by eating something, they take it directly from another organism. And so they hitchhike on other hardworking relatives. And so this is called parasitism. It's basically where one thing grows on another. So you can get examples of parasitism in plants. Ivy and mistletoe are parasite plants because they grow off other plants and drain kind of resources from them. But we're focusing, or my focus is on the microscopic parasites, the things that live on and in you and capture energy from you that way. The other bit of kind of very basics of biology 101 that I just want to cover is how human cells work. But first of all, I want to acknowledge both Ash and the British Society of Immunology. Ash drew all of the pictures for the book, thanks to a grant from the British Society of Immunology. And these great pictures are up on the website that I put at the beginning, and they're out there to share and to use as however we want. And I think they're amazing, brilliantly bright, colourful and cool. But thinking about the cells, all of the information that makes up a cell is stored in the DNA. And the DNA acts as a lending library. So the information that makes you, and not, say, a blobfish, is in your DNA. And that information is then passed by a molecule called RNA. So the information is kept in the lending library, but the cell can borrow copies of it in a form called RNA. And this RNA form is then moved from the cell to little tiny protein factories called the ribosomes. And the ribosomes make proteins. And so you may have heard of this process in the context of the Pfizer-BioNTech vaccine, which uses RNA to lead to a protein. It's the same process that cells do, that we are utilizing to protect ourselves against infectious diseases. And so the process of moving information from the nucleus into the cell is called transcription. And the process of moving from the RNA to make it into a protein is called translation. And I'm saying this because it's this kind of path that the viruses and the bacteria tend to have to interact with and take resources from to make copies of themselves. So that's on the host side, the human side. That's the kind of thing that we're trying to protect. But what about the things that are kind of trying to attack us? So the first things I'm going to talk about the sort of four big families of parasites or pathogens that infect us. So there are four-ish types of infectious agents, and I apologize if you're a hardcore into one bit of this and there's a kind of bit that I'm not covering, but by and large, there's four types. The first are the bacteria, and these are very diverse families. They can live independently, and we tend to treat them with antibiotics, and I'll talk about those later. So these are kind of independent organisms that then can get into our body. Most bacteria don't care or don't need humans to replicate. The most, they live in soil, or they live in spoil pipes from volcanoes, or all over the place. There's lots of millions, billions and trillions of bacteria everywhere. A small number of them can infect you. The second type is the viruses. And the difference between the viruses and bacteria, fundamentally in kind of infection terms, is that the viruses are obligate parasites. They have to infect you to get into your body. They have to steal your resources to make copy of themselves. And examples of viruses include things like HIV, SARS-CoV-2, influenza. And so that's the viruses. The third type are the fungi, and a lot of these are harmless and or mostly harmless, unless the person infected them with is immunocompromised, and then they can kind of get a foothold and infect you. And these kind of cause things like athlete's foot. And finally, there are the eukaryotic parasites, and these can be worms or also single cellular organisms like malaria. And these are complex, multi-stage organisms, difficult life cycles, can often live maybe in one organisms like the malaria lives in the mosquito and then moves into humans and then back again. So much more complicated. The viruses are kind of the most simple things, and then they kind of go up in complexity. So viruses to bacteria to fungi to eukaryotic parasites. And until about 300 or 400 years ago, nobody knew that any of these exist. And the reason why we know about them is because of Antony von Leeuwenhoek. So he was a 17th century Dutch cloth merchant, and he was interested in the quality of his thread. And he developed a microscope, the first microscope, as a way to look and see how his thread was. And when he did this, he opened up this amazing microscopic world, a microorganism world. And it showed that there are other things living than you can see. And this is the start of our kind of understanding of microbiology. But he didn't necessarily link the microorganisms to infection. What was then required was people like Louis Pasteur. So Louis Pasteur was demonstrated that bacteria don't just come from nowhere. So there was a kind of idea that maybe infection was spontaneously occurring. And he explored this in the context of beetroot wine, which I have no idea why you would look at beetroot wine, which you can only imagine is pretty revolting even when it's not spoiled. But there was a problem with it spoiling. And he demonstrated that these bacterial infections could get in and spoil the wine. And his demonstration was using this flask below, which he sterilized by heating it up and then sealed it from the air. And he showed that no bacteria could get into the flask until you broke this kind of neck and allowed the bacteria in. So he demonstrated that infections don't just come from nowhere. They do have to seed from somewhere else. The other sort of key founder of the field of microbiology is Robert Koch. And Robert Koch's key contribution was a set of postulates or rules that can be applied to all infectious diseases. The first is that the infectious agent must be found in people with the disease. Second one is that it must be possible to isolate the microorganism. The third is the microorganism must cause disease when introduced into a new person. And the fourth is that you then have to be able to re-isolate it again. And so it's just a kind of pathway to thinking about it. And I just sort of put it in a different context, was to think about SARS-CoV-2 and 5G phone masts. So you may remember about 18 months ago, there was some very odd discussion that suggested that COVID was caused by 5G phone masts. But if you step back and apply Koch's postulates to it, you can find that SARS-CoV-2, which is this virus here, this is isolated from the nose of somebody with COVID infection. So you can isolate the agent, which is the Koch's first postulate, but you can get COVID in places where there are no 5G masks. So the kind of the underpinning rules that there has to be an agent, it has to pass from person to person, you have to be able to re-isolate it, is very clearly shown for the virus and not for 5G phone masks or whatever, or cats or whatever else kind of people apply towards diseases. But the problem is that a lot of these pathogens are too small to see. So it's very hard to identify them, even with the follow-up to Leeuwenhoek's microscope. It's quite hard to see them. And this is where jelly comes in. And jelly may seem like an unlikely hero in the fight against infectious diseases, but actually a key breakthrough came at a garden party. And this was where a lady called Fanny Hess comes in. Her husband worked for Robert Koch, and she was holding a tea party. And famously, her puddings didn't melt. That's a slight spoiler of my slides going up on me. And so the puddings were made from agar rather than cow gelatin. And because they made agar, they held to a better temperature. And Hess then realized that you could use this to stabilize the types of things that you grow bacteria on. And it's because of this that we get agar plates. And this is from Sophie in my lab has grown some bacteria. Each spot on here came from an individual bacteria. And what that shows is that if you can count the number of bacteria in a sample because you can use this stabilized jelly medium. So bacteria can be counted thanks to jelly, but spreading a sample on an agar plate is effective bacteria, but doesn't work for all pathogens. And as mentioned, viruses cannot grow by themselves. They need another organism or another cell to infect. And a real breakthrough in understanding or be able to propagate, grow viruses to high levels came with the development of things called cell lines. And one of the most famous of these is called the HeLa cell, which is short for Henrietta Lacks, who was an African-American woman born in the USA in 1920. And unfortunately, in 1950, she received news she had cervical cancer, and her doctor took a biopsy from a mass on her cervix. Treatment being what it was in the 50s, unfortunately, she died of this. But unlike most of us, Henrietta's story doesn't end with her death. And unbeknown to her, Dr. Jones passed some of her biopsy to a colleague called George Otto Gay. And this is a sort of complex. Ethically, it was probably the standard of practice at the time. It obviously wouldn't be done now. And there's a lot of kind of work about or thinking about where this is and her importance or Henrietta Lacks's importance in the kind of scientific story. But what Gay observed was that the cells collected from her could be kept alive far longer than any other cell. And he took a single cell from the biopsy and isolated it and allowed it to multiply until he had a uniform population made from this cell. And this one cell that he then grew up you're able to then grow viruses en masse. So you could take a single cell, you can take a flask or a kind of a glass bottle of these cells, and you can grow more virus in them. So you can start counting and isolating your viruses. One of the fascinating kind of things is that because HeLa cells are so widely used, one source has estimated that 50 billion kilograms of HeLa cells have been grown since 1950, which, if it's true, is approximately the size of 1/8 of the whole human population. So from this single person. So these have been a really important tool in understanding how viruses spread, how to grow them, how to prepare them for other things. So that's the kind of agents that we're thinking about, the things that infect us and cause diseases. And pathogens are everywhere. So I want everyone to do an exercise. So I want everyone who's listening online to take a big, deep breath in. So what happens when you breathe in? So you, on average, breathe in about 1/2 liter of air. And you do this about 20 times a minute. If you condense your breath over 1 minute, you can recover about 100 microliters of fluid. So if you breathe all of those 20 breaths into it like a cold fridge, you'd get about 1/100th of an ice cube back. And if you then took your 100th of an ice cube and spread it on one of those bacterial plates that we were talking about earlier, you can recover about 110,000 bacteria. So every time we breathe in, you're breathing in about 10 to 1,000 bacteria. And it's not just bacteria, you're breathing in viruses, you're breathing in fungi. Some estimates put it at that you breathe in 1 million microorganisms a day. And the really important question is, why aren't you sick all the time? Well, if you're breathing all of this stuff in all of the time, why aren't you sick all the time? And this is where the bestology comes in, which is immunology. And well, I say it's immunology. Other ologists may disagree, but it studies how we're defended against infection. So this is a kind of brief schematic of what happens. So if you can see at the top of the slide here, we've got lots of little gray viruses, and they bind to the surface of our cells. So this represents an airway. So this is us breathing in some virus. The virus gets into the cells, and for the virus, the ideal thing would be to replicate, to make copies of itself. But luckily, we have 2 waves of immune cells that protect us. The first are called innate immune cells, and I'll talk about these in a minute, but they come early in the response. So in the first kind of 3 days, they crash into the area and they eat as much of the virus as they can. They also train up the second part of the immune response, which is called the adaptive immune response. So the innate is enough to kind of contain the infection, and then the adaptive helps you clear it up. And this adaptive immunity is really important when we think about immune memory. And I'll talk about the different cells a bit more later. The reason we know we have these white cells is thanks to a Russian scientist called Eli Mechnikov. And Mechnikov coined the word immunology from the Latin word to mean immunis, which means exempt. He investigated the role that white blood cells play in the body. He worked on starfish, not anemones, as I got wrong earlier the other day. And history doesn't really explain why he was working on starfish, but he showed that when you stick a thorn into a starfish larval cells, starfish larvae, other cells gather at the site of the infection. And if you take those cells away, you can show that they're able to eat bacteria. And he called these cells macrophages. And this was really the kind of founding part of our understanding of the immune system. And so I said I'd talk about the white blood cells. We have red blood cells and white blood cells. The white blood cells are the ones that are involved in the immune response. And immunologists like to categorize things into smaller and smaller boxes, which makes it harder for anyone else to understand it. So we can kind of keep our turf to ourselves. But to try and simplify it, I've broken it down into two areas. The first is the innate immune cells. And the two kind of really important ones in this are called macrophages and neutrophils. The macrophages eat other stuff. So they mostly, in terms of infections, they eat bacteria. But actually, if you take a sample of macrophages from somebody's lung and had a look at them down the microscope, you'll find that they're soot black because a lot of them are eating all of the kind of coal and dust and stuff we breathe in every day. And so, especially if you live near London like me, your macrophages are just going to be full of things that they've eaten during their life. The other innate immune cell is one called a neutrophil. And these neutrophils protect you from bacterial infections. Instead of eating things, they contain lots of poison. So they get to the site where the bacteria is infecting, and they dump all the poison onto the bacteria, which kills it. So if you have a splinter in your hand or a spot and you squeeze that spot, not that I'm recommending doing that, all of that kind of white gloop that comes out when you squeeze it is neutrophils and maybe a bit of macrophages. The other family of immune cells are the adaptive immune cells, and these are called the T cells and the B cells. The T cells are really important in like orchestrating your immune response. So if you think about the different pathogens, some like viruses are really tiny and get into your cells and live in your cells, whereas others like parasitic worms can grow to as long as like 20 foot. And so the strategy your body uses to clear the different things has to be different. And the T cells kind of are like the general. They decide what's going to happen and how to clear the infection. Whereas the B cells are ones that make something called antibody. And so probably one of the words that's come up a lot in discussion in the last 18 months is antibody. And antibody is this amazing biological molecule that can recognize one thing very, very specifically. So a B cell that recognizes SARS spike protein is able to make an antibody that will bind just to the surface of the SARS virus and not to, say, to the influenza virus. So these are really important in kind of protecting you both at the time of the infection and then if you get infected again. And your immune system protects you during primary infection, but actually it stops you getting sick again. The reason why small children, babies, get loads more colds than adults do is because our body sees them and we get trained and we prevent future infection. So once the immune system has recognized and it responded to an antigen and an infection, it exhibits a characteristic called memory. And these memory responses are characterized by faster and stronger. So basically, if you see flu today, you might get sick and you then recover. But if you see it again 2 weeks later, your immune system is prepared and it can fight it off much better and much faster. So basically, most of the time, you won't get infected by something that you've seen before. And it's this idea that's the basis of vaccines. And the immune memory can be incredibly long-lived. And there's a great story about a measles outbreak, which kind of puts it into context. So the Faroe Islands are a long, long way north of Scotland. And in the 18th century, they were basically isolated because it's so far away. And in 1781, there was a measles epidemic in the Faroes. So there was one measles epidemic and a lot of people got sick, but most people got recovered. And then there wasn't much change over in the people, and there were no further outbreaks till 1846, till 60 years later. When the 1846 outbreak happened, not a single person who'd survived from 1781 was infected in 1846. So this kind of this memory of the infection protected the elderly people who left in the island because they'd been infected 60 years earlier. I've also remembered that you can let go of the breath that I asked you to do about 6 slides ago, because you might be feeling a bit faint by now. So immunology really helps us to understand what happens in one person, but you need more than one person for outbreaks to occur. The epidemics that we have only started occurring once we had cities. So the Justinian plague, which occurred around the time of the Romans, was probably the first major recorded epidemic. And it happened because there were cities and also transit of movement of people between the cities. And so modeling how disease spreads is vital in controlling these outbreaks. And the kind of basis of this modeling came from 1854. So in 1854, there was a cholera outbreak in London. It emerged explosively onto the streets of London. It wasn't particularly uncommon, but what was different was John Snow, a doctor, tracked where all of the cases of the cholera were happening and then drew a map and tried to work out what was in common. And what was in common was they were all using a single water source, a single pump to get their water from. This pump was getting infected by polluted sewage water, and people were basically drinking sewage water from the pump, except for famously the people who worked in the brewery who got, so that if you can see, there's a not very clear, but there's a white box where there are no infections. And this is because it was a brewery, people drank beer there instead of water from the dirty pump. So the lessons are that a single source of infection can spread things, and that if in doubt, drink beer instead of water. But Snow wasn't the only person who kind of developed epidemiology. Another person who you may think of more in the context of nursing, but actually was really important in epidemiology, is Florence Nightingale. So she's kind of famously remembered as the Lady of the Lamp for her service in the military hospital in Scutari during the Crimean War. But actually, one of the things that she developed afterwards was a thing called the Nightingale Rose diagram. And she recorded the causes of death in soldiers, in British Army soldiers in India. And what she could see was that there were patterns in the types of death they had and when the infection spread. And it was kind of linking, what Snow did was linking spatially infection, and where Nightingale was looking at temporally, how infection spread over time. So it was these kind of underpinning bits of understanding that then helped us to think about how diseases spread. And the models have developed and got more and more sophisticated. But one of the kind of key ideas that has occurred or sort of, again, has cropped up a lot during the COVID pandemic is this idea called the R0, which is for some reason has been abbreviated to R. But R0 is the number of people that get infected. So if this idea works better if I'm standing with an audience in front of me. But if I was infected and I sneeze on the front audience onto two people, and the R0 is 2, those 2 people will get infected. If they then turn around and sneeze behind them, if their R0 is 2 as well, they'll infect 4 people. So R0 enables things to double. So, or whatever the R0 is, it spreads. It's the number of infected people from a single point source. And at 2, things can double very quickly. You don't have to have a very high R0 value for things to double or to increase in size very fast. So this is a demonstration of the kind of famous fable of a man who was asked to be paid in rice as doubling amounts of rice for each square of a chessboard. And it sounds like a trivial amount of rice because you start at 1, you get 2, 4, 8. By the time you get to the sort of penultimate row, the pile of rice would reach up to the sun. And on the very last row, it reaches out to Jupiter. So doubling happens very, very fast. And this is why with the SARS pandemic, when the R0 has got anywhere above 1, things have spread very quickly, especially when there's a lot of people infected already. The other kind of visual model to think about this is the Lillypad model. And a lot of doubling can happen before the spread's widely visible, especially if many of the cases are without symptoms. And so we think about this in terms of a lily pad. And the question that I'm going to put to you is, if the lily pads double every day, if you start with 1 lily pad and it doubles every day, then the question is, if the lily pads cover the pond completely after 36 days, how many days would it take to cover half the pond? So I'll leave that with you, and it kind of, you have to think about that, or you have to buy the book because it's got the answer in it. So moving on to viral transmission in the context of these kind of doubling things, is that this is a model from Imperial, and it shows something interesting, is that things move in and out of control. The difference between SARS-CoV-2, which is up here, this one saying COVID, and SARS-1 is that SARS-1 infected people after people were symptomatic. So if you had SARS, you tended to go to hospital and then you could be isolated. Whereas with SARS-2, there are lots and lots of people who've been infected, but actually are asymptomatic and spreading it. So diseases that infect people without them being seen spread much faster than diseases that infect people and then lead to symptoms. So this is where you need tools like diagnostics. So diagnostics help us know who's been infected. And this shows the amazing speed and development of diagnostics in the UK. So it went from the early outbreaks where there were not very many diagnoses being made a day to a point somewhere in the middle of March 2021 when 1.9 million diagnoses of COVID were made in a single day, or diagnostic tests were conducted, not diagnosis. That's way more. So look, that nearly 2 million tests were done in a single day. And it's this ability to track the virus, to understand how it's spreading, that then helps us to kind of move or change behaviors and understand what's happening. And the diagnostics were really done by two forms. The first was the lateral flow tests. And to do this, to recognize kind of infections, we use the power of the human immune system, particularly antibodies. So as I said, antibodies can bind things, and they can specifically bind a single thing. And the lateral flow test is actually based on the pregnancy test. It has the same kind of thing. So you start off by putting your sample. Those of you who've done these will know you kind of drop your snotty bogey. Once you've stuck your thing up your nose, it spreads up the tube. The first T is just to show that the test is working and that there's liquid in there. And that binds some antibodies. The second one, it binds antibodies that are specific to the virus and the virus particles that were up in your nose. The second test that have been used is the PCR, polymerase chain reactions. And I've just put this in here as a kind of bit of historical interest. PCR machines don't look like this now, but you have to change temperatures to make PCR work. And what it used to be is that somebody used to have to stand there with the sample in one bucket and then lift it out and put it in the next bucket and then lift it and put it in the next. It's now moved to where you can do it on a single chip that you can plug into your phone. And what the PCR does is it's an amplification process where you can basically extract the DNA or RNA from a virus, and you can measure how much of your virus is in it. So those two approaches were kind of the workhorse of the of how we measured the spread of the disease. So that's the end of part one. So I've talked about microbiology, a bit about immunology, a bit about epidemiology, and a bit about diagnostics. And now I'm going to move on to part two, which is thinking about the solutions, how we prevent and cure diseases. So the simplest way to stop infections is to never get them in the first place. And we've seen the clear impact of non-pharmaceutical interventions, or NPI, so basically things that don't involve drugs, in stopping spread. The lockdowns, the washing hands, the mask wearing, all have stopped spreads. Hand washing is a really important process. It shows to reduce the transmission of respiratory infections by 20%. However, and here's a gross fact, prior to COVID-19 in the UK, the frequency of hand washing was embarrassingly low. One study reported that only 32% of men washed their hands after going to the toilet in a motorway service station. Thankfully, the percentage did increase during the 2020s. And one of the kind of weird things I noticed was I went to a concert, and there was a queue at the gents' toilet, which was kind of unusual. And it wasn't a queue for the toilets, it was a queue for the sinks afterwards, which makes you kind of think, what's happening normally if there are no queues and men aren't necessarily washing their hands? But thinking about hand washing, amazingly, hand washing wasn't always seen as the right thing to do. Prior to the 1860s, surgeons were not the methodically clean individuals we think of today. And they took pride in the number of bloodstains on their surgical gowns as a kind of mark of operations that had gone well. And unsurprisingly, this meant that post-operative infections were very high indeed. So if you went into a surgeon, you're pretty much likely to get infected and die as of the thing that you went in for. However, Ignaz Semmelweis, a Hungarian doctor working in Vienna, first challenged this dogma, and he linked the practice. So what used to happen was people used to examine corpses in the morning and then go and examine pregnant women in the afternoon. And he linked this kind of touching dead bodies before touching pregnant women to a high rate of death or fever in both the mothers and the children after birth. And by introducing a policy of hand washing, he led to a massive reduction in this. But sadly, the establishment at the time rejected those ideas. And once he passed away, the practice did not catch on until the work of a second surgeon called Joseph Lister. Lister developed or built on Semmelweis' ideas, but also on Pasteur. So because Pasteur had said there are these bacteria that can infect you, he had this kind of solid grounding with which to kind of understand and think about how you can prevent it. And he used a solution of carbolic acid and sort of basically tar, which killed the bacteria and prevented further infections. And it's kind of from him and from his name that that Listerine comes from. So it's thanks to kind of Semmelweis we have these kind of preventative strategies. And prevention is much better than cure. So thinking right back to the beginning of the talk where I talked about the increase in lifespan, one of the big things has been the increase in access to clean water. But the other important thing is vaccines. And vaccines are amazing. Vaccines work by training the immune system to recognize and remember the infection without ever getting infected. I talked about immune memory earlier, about how the people living on the Faroe Islands had seen the virus and were protected against future infections. The difference with vaccines is you don't have to get infected. You don't have to get measles and run the risk of dying. You can just have the safe memory without ever getting sick. And just to show you how well they work, here are a number of different infections. And what you can see is an arrow every time the vaccine was introduced. So there's an arrow here, the vaccine's introduced here, the vaccine's introduced here, the vaccine's introduced here. What all of these have is a massive crash in the number of cases post the introduction of the vaccine. The vaccines prevent the infectious diseases, preventing disability, disease, loss of life. They're an incredibly important tool. And the vaccine aims to do it works on like 3 levels. The aim is like immunologically is to make an antibody response that binds and kills the virus or the bacteria. But they also work health-wise. They work to prevent diseases. So yes, it's great to stop the infection, but if you can't control an infection, you really want to stop the disease so that if you do catch the virus or the bacteria, you don't get properly sick from it. And the third thing that they do, and this is their real power, is that they stop the disease spreading further. So normally thinking about my talking about that R0 and the idea of things spreading, what would happen is I get infected, I pass it to somebody else on the tube, they get infected, they pass it to somebody else on the bus, and it spreads and it spreads and it spreads. In the case of herd immunity from vaccines, if you get infected and the person if I get infected and go and sit on the tube, I don't know I'm infected, but I sit next to somebody who's already been vaccinated, they don't get infected. And then it doesn't matter what happens to the person sitting on the bus, because the person who sat next to me never got the infection in the first place. So this stops the infection spreading any further. So these vaccines are incredibly useful and potent tools to control diseases. And I'm just slightly running out of time, so I'm going to skip ahead of that one. We trace vaccination back to two people right turn of the sort of 18th, 19th century. First is Lady Mary Wortley Montagu, who was a smallpox survivor. And she introduced this practice called variolation to the UK, which is basically blowing the scabs from smallpox sufferers up the noses of children to stop them getting smallpox in the first place. It was effective, but it was not necessarily that safe, because sometimes you blow too many scabs up the nose or the person would be more susceptible. So she introduced the idea that you could prevent diseases by exposing to another infection. But it was actually Jenner who then developed the true vaccination. And the story goes that he had noticed that milkmaids, because they're exposed to cows and cowpox, tended to have beautiful skin, better, cleaner skin, because they didn't have smallpox infections. And so he isolated cowpox from an infected cow, and then he he injected a boy with it, and then exposed the boy to smallpox. And the boy, Phipps, was protected against smallpox. And this showed that you could use one organism to protect against another. The real nub of the story is slightly more complicated, and hence that's why you need to buy my book. Another subtle plug. And smallpox has been eradicated. This is a picture that shows you the power of smallpox vaccination. These are twins. The boy on the right got vaccinated. The boy on the left was not vaccinated. They're obviously both exposed to smallpox. This boy on my left, it has the smallpox infection. So you can see how how horrible the disease was and how protective the vaccines were. And smallpox eradication is one of the amazing medical breakthroughs of the 20th century. By 1979, nobody was infected with smallpox. So this is the last person ever to be infected with natural smallpox. And since the eradication in 1979, nearly 300 million people died in the 20th century of smallpox. If you take that and the size of the population and expand it forward, huge numbers of people have been saved. But those huge numbers are quite difficult to think of. So I like to think of it a different way. Since 1979 and the eradication of smallpox, more people have died of the following things: death by sacrificial goat, crushed by a cactus, killed by a broken golf club during a backswing, through excessive use of deodorant spray, riding a Segway over a cliff, whipped cream explosion, or my absolute favorite thing that's killed more people than smallpox since 1979, the atomic wedgie. And so that's just incredible. All of these stupid deaths have actually killed more people than smallpox, because nobody has caught smallpox since 1979. It's just a huge, incredible achievement. And we are so close to doing this with another disease, polio. And to develop the polio vaccine, we owe it to two scientists who worked in kind of competition at the time. They were called Jonas Salk and Albert Sabin. And they had two opposing views of how to make a vaccine. Salk preferred the inactivated virus and Sabin an attenuated one. So basically, one was like, we should kill it with chemicals, and the other one was we should grow a weak version of it. And Salk's method was kind of quicker, and he did the first successful trial to show that it worked. And this was a huge success, and he was declared a miracle worker by nearly everyone, except for Sabin, who described him as a mere kitchen chemist. And they kind of had a real set two together. But between them, they've developed these polio vaccines, which are leading to the eradication of polio. And we're so, so close to that happening. Another hero of immunology, and this is probably, to me, the most important person that you've probably never heard of. He's called Maurice Hillman. He spent most of his career working for Merck. And between 1957 and 1984, he and his team developed 14 new vaccines, 8 of which are still in use today. So there's a huge input of what he's done to prevent and protect against infectious diseases. One of the vaccines is the one for mumps. So this is part of the MMR vaccine. Mumps virus causes infection or swelling of salivary glands. It can lead to a viral meningitis, so it can lead to disease of the brain. It's extremely uncommon now, thanks to Hillman's vaccine. And the vaccine's named Geraldine because he isolated the virus from his daughter's throat. So this is Hillman's daughter, Geraldine, who he got the virus from. And then this is his younger daughter being vaccinated with the virus that came from her throat swab. So this is kind of very much keeping it in the family. And I know we're kind of meant to show pictures of vaccines and people looking happy, but I just like the idea of her looking on as her sister is slightly in pain because of what she did. And we have so many vaccines available. We've gone from the 1800s, we have vaccines against bacteria, vaccines against viruses. As technology has developed, as our understanding of pathogens has developed, we've got different ways of building new vaccines. And then in 2007, this is where I started lecturing about vaccines. And there was a kind of feeling at the time that there were like vaccine development had ended. There was nothing new going to happen. It was kind of seen possibly as a dead end. And then actually we've got 6 new incredible vaccines since then. So whether that's correlation or causation of me starting working on vaccination, I'll leave that to you to decide. And the big one that will all be at the front of your mind now is the COVID vaccines. And so the COVID vaccines aim to do one thing, which is to target this protein on the surface called the spike protein. And spike is how the viruses get into our cells. And so if you can make something that stops the spike getting into the cells, and basically the vaccine makes you make antibodies that cover up the spike protein and stops it getting into you, you can stop infections and you can stop it making severe disease. And there have been lots and lots and lots of different approaches to COVID vaccines, but really 2 have kind of the ones that are used in the UK and very widely globally. And they're the mRNA one and the recombinant, the viral vectored one. So just to go a bit more into detail about those ones, the first one is the Oxford vaccine, and this is called ChAdOx-nCoV-19, and it was developed by the team at Oxford University, which is where the Ox bit of ChAdOx comes from, led by Dame Sarah Gilbert, and what it does is it uses a weakened virus to deliver a genetic cargo. It basically, it gets into your cells make a tiny bit of SARS-CoV-2, not an infectious bit, and that's enough to train your immune system so you don't get infected before. The second type of virus is the mRNA virus. And this is the one that's Pfizer, the Mrna one. And one of the vital breakthroughs came from the work of Dr. Kati Kariko. Kati's an amazing, inspirational person. She fled communist Russia in 1985 with $1,200 hidden in a teddy bear. And she realized that the way that the cells see foreign RNA, could be stopping the way that the vaccines work. So if you inject some RNA into somebody, it doesn't make a protein, so you don't make the immune response. And she chemically altered the RNA slightly, so your immune system doesn't recognize it quite so much, and you actually make a much better immune response against the protein. So thanks to her insight, we have these incredible RNA vaccines that are going to protect us against not only COVID-2, but probably flu, RSV, lots and lots of things in the future. But it's not just about vaccines. Sometimes you do get infected. And despite our best preventative strategies, we need things to stop those infections. And luckily, drugs do exist that can cure these infections, both viral, bacterial, fungal. And thinking about antibiotics, these are drugs that prevent or treat bacterial infections. And I work next to the lab where Alexander Fleming made his breakthrough. And his story has been often told. Basically, he went away on holiday. His lab was a total mess. And he came back and actually found somehow that there was a mold infecting his plates that killed the bacteria. We've tried to repeat this kind of breakthrough in our labs by leaving it as a constant mess. And yet we've got no Nobel Prizes yet. But maybe tomorrow's the next day. So he identified this mold that could kill bacteria, but the story doesn't end with Fleming. The fungi contaminating his plates was a penicillium, which is where the compound penicillin comes from. But this discovery was a long way from the actual drug. It actually took another two scientists called Flory and Chain, and Chain later worked at Imperial College as well. They did these discoveries at Oxford, it pains me to admit, and they developed a process for extracting penicillin from fungal broth. And they shared the Nobel Prize in 1945 with Fleming for this discovery. So they showed that you could extract penicillin from penicillium mold, but not really in large enough volumes to be able to produce the antibodies that we need, antibiotics that we need. And this is where moldy melons come in. So Mary Hunt, who's the lady on the left, not the melon, was working for the US Department of Agriculture in Illinois. And she had the romantic job of searching local markets to find better strains of mold. And in 1943, she did indeed strike mold. She found a strain of penicillin that on the skin of a going off cantaloupe, which produced 200 times as much penicillin as the original Fleming strain. And it was from this strain, which was then kind of farmed and grown up like a bit like they kind of make better cows, select for better cows, that enabled them to make enough doses to have 2.3 million doses in time for the D-Day landing. So Fleming's role was very important, but you also need Chain, and you also need Flory, and you also need Mary Hunt, and all the team that kind of developed this into an industrial process to get to having penicillin. Penicillin was the first of the kind of modern antibiotics, and then from there we got other antibiotics. And I'm just going to mention the sort of golden era of antibiotics. And Selman Waxman, who is a Ukrainian-born microbiologist, spearheaded the work in this kind of golden era of antibiotics. And I mentioned where he works, which was Rutgers, and this is dear to my heart because it's where I began and ended my international rugby career. If you look very closely at the photo, you might be able to spot me. Admittedly, only 15 people at the college out of 67,000 students play rugby. So it wasn't quite the selective pressure as maybe at other places. But Waxman was really important in terms of antibiotic development. He actually coined the phrase antibiotic, and his lab generated many of the antibiotics still in use today. In 1943, Waxman and particularly somebody called Albert Schatz in his lab was testing different compounds for their effect on the growth of E. coli. And they actually tried to replicate Fleming's accidental approach by isolating molds from mud, which could kill bacteria. And they found another compound called streptomycin, which was able to kill TB. So he developed kind of the next generation of antibiotics. The problem is, unfortunately, the bugs are fighting back. So antibiotics, bacteria are beginning to grow resistance to antibiotics. And there is a kind of developing pandemic within kind of healthcare of these bugs that are no longer able to be killed by antibiotics. So what this kind of shows is that basically every time a antibiotic has been identified, there are bacteria that have grown back, have been able to grow that can resist it. So 1940, streptomycin was identified. By the 1960s, there were antibiotic-resistant, streptomycin-resistant bacteria. So there is a kind of ongoing battle between both the bacteria and the drugs that is kind of needs research and development to keep going. But antibiotics don't work on all pathogens, particularly not to viruses. Nearly 30 years ago, in November 1991, Freddie Mercury died of AIDS, which is the disease caused by the virus HIV. At the time, the virus was untreatable, and HIV infection was often described as a death sentence. However, in the 30 years since that day, antiviral drugs have completely changed the face of HIV infection and treatment. And where the treatments are used and available, it's now a chronic condition that can be managed into old age. And we've seen astounding successes in antiviral drugs. And part of the reasons for this is because of people who then looked at the viruses. So whilst AIDS sort of emerged in the 80s, it wasn't known what was causing it. And it was down to these three scientists, Gallo, Sinosi, and Montagnier, who had identified the HIV virus, the virus that gets into your T cells, damages your immune system, and then causes the disease. And by identifying the virus, they then opened the door for another very important scientist, Trudy Elliam, to then develop drugs to treat it. So she's the kind of grand dame of antiviral drugs. She graduated in chemistry in 1937, but as a female scientist, she was unable to find a research job. So she had to save up to do her master's degree part-time while working as a school teacher. She was then excluded from doing a PhD because she was a woman. But luckily for the world, she got a job working at Wellcome in 1944, developing antiviral drugs. And it was thanks to her and another scientist, George Hitchens, who developed the first anti-HIV drug, which was then kind of built into a range of kind of treatments to prevent infection. So they kind of all work together to lead us to this point where we have these treatments that work. And we have drugs against bacteria, we have these antiviral drugs. We also have drugs against the eukaryotic parasites. And so you might wonder what these two things have in common, what the malaria-carrying mosquito and gin and tonic have in common. So malaria causes a disease characterized by both fever and tiredness, and it's driven by the destruction of red blood cells by the malaria parasite. But it was discovered a long time ago that it was bark from a tree called the quina tree protected against malaria infection. And in the 1820s, two French scientists discovered that actually there was a single compound called quinine within this bark that could be used as a prophylactic drug. And British colonists stationed in India were required to take quinine in some form. And they discovered that if you mixed it with sugar and fizzy water, it made it slightly more palatable, especially if you added gin. And so this is where the sort of gin and tonic came from. It was an anti-malarial treatment. Unfortunately, a bit like the antibacterial drugs, quinine started to fail and new drugs were needed. And this is where UU2 comes in, who led a project to explore new anti-malarial drugs. And in her attempts to develop the drugs against malaria, she went back to traditional Chinese medicine. Between 1969 and 1971, they tested 2,000 traditional Chinese recipes. But she kept on going until she found a recipe in a book called Emergency Prescriptions Kept Up One's Sleeves by somebody called Ji Hong, which involves soaking leaves from a certain tree in water. And she tweaked an extraction method to isolate the drug called artemisinin. And this artemisinin has saved millions of people. So it's sort of another way of discovering anti-pathogen drugs. So I've talked a bit about the underpinning science, a bit about where we are. So just a little bit on thinking about the future. So what things might help us protect us against infectious diseases in the future? So there's a number of things. The first is gene therapy. I think there's a new technology called CRISPR-Cas, which is reaching into all stages, all phases of biological life. And it may well change how we prevent infections, how we treat people who maybe are more at risk of infections. I think we're likely to have better diagnostics. This is old Bones McCoy using his tricorder. I think devices will get better and faster at measuring where infections are happening. We're definitely going to have better vaccines. We've seen that this year. I think the kind of explosion of vaccines this year will lead on to new things. We may well have ways to manipulate the bacteria that live with us called the microbiome. We may think about ways that infection links to other conditions. I'm getting close to the end. And we will have other better therapeutic antibodies. So we will harness the immune system in better ways. So there's two ways of looking at this. Pasteur is the kind of from the school to say the microbes will have the last word. And then another scientist, not William Stewart, but this is a quote misattributed to him, said, it's time to close the book on infectious disease and declare the war on pestilence won. And we're somewhere in the middle. The microbes are fighting back, and yet we are making much progress. And I'll just finish up on this last image, which is from slightly more than 100 years ago. And this is the Spanish influenza outbreak, which killed 100 million people, so 10 times as many people as the First World War. And it was caused by a disease the cause wasn't known. They didn't know that there were viruses or human viruses, that the influenza virus existed, and there were no drugs, there were no vaccines. And we've had a similar outbreak 100 years later in a much bigger, much more interconnected world. And yes, people have died, but the percentage of the world is much, much smaller. So I think there is hope for where we're going. And just as I finish, I want to say thank you to Caroline Hardman and all the team at Hardman Swainson for all their help in the book, to Sam Carter, my editor, and all the team at One World, particularly Holly, Kate, Rita, and Lucy, who made it such a beautiful thing, to Dan for putting me on the path in the first place, Ash for all her beautiful pictures, and the British Society for Immunology for funding them, and then all my friends and family who've just supported me this time. Thank you.